Jessica Christoph is Vice President of Research and Development at Phylos Biosciences, a leading company in the genetics and biotechnology of hemp and cannabis. Jessica was one of the first scientists at Phylos and came to the company after working for a few years as a staff scientist at Synthetic Genomics. Jessica is an Oregon State University graduate and will be talking today about the genetic research currently at Phylos. Dr. Alicia Holloway is Vice President of Data Science at Phylos. She was an assistant professor at UC San Francisco School of Medicine and was the founding director of the Gladstone Institute's Bioinformatics Core Facility. She earned a PhD focused on molecular evolution at the University of Texas in Austin. Today, Dr. Holloway is going to discuss her pioneering work on understanding cannabis genetics and population biology.
We're actually going to do this together because a lot of the work that we do at Phylos is a team effort, and you'll see that in all that we talk about today. So I'm going to start by introducing, you guys kind of saw a snippet of this before, what we started with when I and the founders started the company in 2014. There wasn't a lot that had been done in cannabis, and specifically there wasn't a lot that had been done in regards to genetics. We had a good draft genome to start from. The molecule had some analysis that had been done. Obviously, the University of Mississippi has a license, so they had been doing some studies, and they had some papers that came out, but there really just wasn't a lot. And so from there, one of the founders, Mowgli Holmes, had recognized that a really big problem that folks had been running into in the cannabis industry is that, if you're a recreational user and you go to purchase something from, say, a dispensary, if you don't get what you thought you were going to get, it's a disappointment. That's really all it is a disappointment. But if you're a medical user, it's a problem, and that's really unacceptable. So we recognized pretty early that we could use genetics to help folks in identifying what they were purchasing. That's a little bit harder than it sounds. It sounded really easy. We were like, I had done a ton of sequencing in the past in previous jobs, and I was like, yeah, I can do this. And we quickly realized that before we did that, we needed to analyze what was out there to then compare each time a new sample came in to what was there and tell them if they had a clone or what they had. So this is the history of Phylos, and it started with the Cannabis Evolution Project, where we worked with growers from around the globe, and we started collecting DNA material that we could sequence. And we did 1,000 different samples, and as you'll see in a bit, it was very effective in giving us that look into what the diversity of cannabis is, maybe cannabis or hemp. That takes time, though. Collecting that many samples takes time. And also, this industry, as I mentioned, while there wasn't a lot of science there, there was hesitation. The industry is not, it has been illegal for a very long time, and they're not used to talking with scientists. And so when we came around and we had said, we'd like to sequence these, we'd like to do this project, there was a lot of hesitation. So it took time to collect those samples. And in the meantime, I and my team developed a sex test that's a basic qPCR test. I'm happy to dive into what the particulars of that are. It's a very effective test. It saves a lot of time, a lot of money, and that was a great thing to introduce us to that community and to show them that we're just scientists creating tools for them to use for a crop that has really not had any work done on it. From there, in 2016, we released the Galaxy. This is a visualization that enables us to look at all of the samples that were collected for the Evolution Project, as well as we continue to sequence to see the samples go up and compare them against that database. In addition, we took the samples and we introduced them to the Open Cannabis Project. Now, the Open Cannabis Project, the focus of this is to take information such as the sequencing information, genomes, and to make them publicly available to scientists other than just us. We feel that's really important, especially for this industry, because this industry needs to grow quickly if it's going to succeed. And the only way we can do that is as scientists to work together to collaborate. And so Phylos and other genetics companies in the field have released to the Open Cannabis Project, and the Open Cannabis Project is actually just a nonprofit that has those conversations with genetics companies to release that information to the community. And our information, which Alicia may touch on later, is all released up to NCBI. So 2017, we released Phylos Certified, which is the genotyping project, or the product that enables you or a patient to give a sample, have it sequenced, and realize what it is. And then Stuff and Things is what we're working on right now. We'll talk about that in a second and just some of the new things we're working towards. So in developing the Galaxy and in collecting this data set, we had to have the discussion of what is the best route to go about this? So we have 1,000 samples. We need to compare them quickly, and then we need to create a test that can reproducibly have high specificity moving forward for every sample from today until years from now. So when we started the project, we used things like GBS and RAD-seq and these kind of exploratory techniques to identify SNPs that covered the entire genome. By collecting these samples that were from around the globe and represented hemp and the drug cultivars, we were able to then select those SNPs that were appropriate for all the diversity that represented cannabis. Finally, we felt that the best way to do this reproducibly was, while you can use GBS as a discovery tool, we went the route of using targeted sequencing as our product tool to provide reproducibility. So right now, I believe we're at 3,600 SNPs that we actually look at, but 2,500 that actually build the Galaxy on a daily basis. This is the Phylos Galaxy. Hopefully we can get the movie working, maybe. So each star represents a different cannabis variety, and there are small stars and there are large stars. And the large stars are actually clone groups. Now, for folks that may not be familiar, cannabis can be cloned, and there are a lot of clones that move around the globe. So there are unique varieties, and there are these clone groups. And so this, and in this video, we're looking at sour diesel. What's nice about the Galaxy in particular is that we are able to not only go to a single star and actually look at that, like I just showed, but we can also look at the clone groups. And this will really show us the difference between the strains, both in their unique, say there's a single star, a single variety that represents sour diesel, versus a clone group. When you click on a clone group, you can actually see all the different names that people have given that, even though genetically it's the same thing. I hope I'm not moving too fast. Is it going to let me out? You guys want to watch the video one more time? Okay. So the Galaxy is really nice. It provides this 3D visualization. Each of these colors represent different populations. There are, in fact, 6, which I'm about to jump into. It's fun to tool around in, that's for sure. But what I really enjoy about the Galaxy is actually the report. So the report enables you to look at a specific strain or varietal, they call them strains in the industry, and better understand it. So the closest genetic relatives, in the center you'll see, I have a mouse here, this is a sour diesel that was being searched. These are siblings, and then so on and so forth. These white circles are actually those clone groups that I mentioned, and if you click on them, you can actually see some of them may all say sour diesel, and they got it right. Or they used our services to find out that they were part of that clone group. Some of them may have something totally different. So this has really empowered people to not only the patient to get consistency in their product, but also for growers to provide that consistency. What's nice about this report as well is it provides more information in regards to genetic novelty, population, variation, and distant relatives, which I'm about to jump into. I would definitely suggest, though, if you guys go to this, you'll see right here there is a little I next to all of these. And this actually provides detailed information on how this report was built, how that particular infographic was built, and the scientists and what they referenced when they were building it. So I want to dive into these three a little bit more. We find that people interact with them a lot, and it makes perfect sense. So obviously, the closest relatives provides a lot of information on what's available, what's there. This is just really helpful for consumers. The genetic variation in particular is very helpful for breeders and growers when they're looking at how many generations am I going to need to backcross to get a stable line, for example. Whereas the population profile, we've broken this up into this is the entire representation of the galaxy, whereas this is just the one strain that you're looking at. So the genotype report provides a lot of information that was never available to this community, and it has really expanded their ability to take the information that they have now on this one strain and grow, or this variety, and grow from it, and either breed or just put it straight into production. This goes back to what I was initially saying, that when we started, it took us some time to actually collect these samples. It took us some time to start as a company, and that was because while the community was very excited about science, they also were a little bit hesitant. So everything that we now do, we try to educate in the process. We try to empower folks so that they think to themselves, I should ask my local scientist. I should reach out and ask for help because maybe there's a tool I can use. And I love this as an example because this is the seal that we put when somebody gets their variety sequenced with us. And you can see that each one is different. Each color is a representation of the 6 populations. And then that spattering are actual SNPs and how they vary between those different things. And so through this very nice imagery, people are able to digest that this isn't, sequencing is unique to these different strains, and of course, unless you have a clone, and that this provides not only consistency for them to see that uniqueness, but consistency again to the consumer to convey that message. So the next step for us is to work with folks, and I apologize, I copied this over from another slide and I don't have my references, but I'm happy to provide those. The next step is to take all of this information and help folks moving forward with breeding. I've mentioned that a lot of them are using this report to better understand their strain. Well, what are they going to do with that information moving forward? Right now, the galaxy does not have phenotypic information associated with it. And you all know that that's very important to them better understanding how the genetics relates, right? And so what Phylos is doing is we're actively working with folks and partnering with them to match that genotypic information with standardized phenotypic information. And I separate out chemotype here, recognizing that it is a phenotype, but for our community, it is such an important phenotype that it is sectioned off. So before I toss it over to Alicia to jump into the next stage of genetic analysis that we're doing and what we're some exciting things that we're starting to learn, some tools that are currently under construction that we're excited about, that we're partnering with folks. I've already mentioned that we're working on better chemical profiling, better phenotyping profiling, genetic analysis for disease identification. We're trying to work on making that SNP pool broader so that it works and matches up nicely with phenotypes. We're working on a lot of different things, and we're definitely open to collaborations. So while we focus on one story in this particular talk, please don't hesitate to reach out because we've got a lot of scientists that build up our team. Alicia.
Hey. All right. Thank you for inviting us. This is exciting to get to talk to an academic audience. This has been my first foray into industry, and what I'm really appreciating about it is that we have all of these different facets of the company, from marketing to finance, that are all driving towards the same goal. And so we are not only in an exciting industry, but we're all working together to make all of this stuff happen. So I'm going to tell you a little bit about some new genomic resources that we're developing. As Jess mentioned, when the company started in 2014, there was not a lot of genomic information. We sequenced a genome and made it public, and we've been working with that genome to develop our genotyping test since then. But then we realized that we really needed to go deeper. We needed better genomic resources, better annotation. We needed population genomic data. And so in the beginning of this year, we started down that road. This is like just the kitchen sink that we're throwing at developing these genomic resources. We're using all of the modern technologies that one would use to get a really high quality genome. And then we are also we have collected population genomic data on 96 samples. We then used Google's deep variant to identify SNPs, and in that we have narrowed down the SNP pool to a very consistent set and have a 25-fold increase in the number of sites that we're interrogating with our new genotyping methods. This will take us really into molecular and genomic breeding and help us to identify genetic markers for different traits. And then today I'm going to talk about one research project of the many that we have going. This is a very collaborative company, and so all of the work that Jess and I talk about really does touch all of the scientists in our company. So I'm going to talk about this cannabinoid synthase work. I think that you all had probably a really nice introduction to cannabinoids and terpenes already, so forgive me for telling you a little bit about what you already know. This figure is from the state of Washington. They looked at the chemical profile, or the THC and CBD of a ton of plants over many different labs. And you can see that they break out into a few different categories. So there are those very high THC, varieties that we call chemotype 1. There are the very high CBD varieties that we call chemotype 3. And then there's this set in the middle that we often refer to as chemotype 2 that have these mixed ratios, but really you can see that that's not explaining, 3 categories isn't really explaining what we see out there. I mean, there's a big difference between a 15-to-1 ratio and a 1-to-15. I mean, these plants are obviously going to be producing very different effects in patients and recreational users. Let me get my water real quick. So since these compounds, they're not the only important thing in cannabis. Obviously, terpenes and other cannabinoids are very important in producing the effects that we see with this plant. But because there's so little research done, we really had to start somewhere. So we wanted to better understand the genetic basis of THC and CBD production. I won't spend a lot of time on this because I think you guys are probably totally up to date on this, but from these precursor molecules, there are at least 2 synthase genes, the THCA synthase and CBDA synthase genes that are used to create the acid forms of these molecules. And so I'll focus on those today. So there's a little bit of research that has tried to understand the genetic architecture of how these cannabinoids are formed, or what genes are involved in forming these cannabinoids. And for a long time, I thought that it was a single locus that had allelic variation that led to the production of CBD or THC. And then with some genetic work and some sequencing, it's become pretty clear that it's really a two-locus model. There's really a CBDA synthase gene and a THCA synthase gene. But then it gets, let's see. So we had this ancestral synthase probably. So this is not found in hop, which is the sister species. So we had an ancestral synthase. It very likely duplicated. When you look at these synthase genes, they're really similar at a genetic level. There are parts of the gene that are identical. I mean, over hundreds of samples from all over the spectrum of cannabis. You look at them, and these particular regions are identical. So it's pretty clear there's been some gene duplication, and then some subfunctionalization that has led to the production of THCA and CBDA. Here's one study that's really nice. This is from George Weeblin's lab, who's been a real pioneer trying to overcome the difficulties of working on cannabis in a university setting. He's been working on this plant for a long time, and I always like to give him credit because he's done such nice work in difficult circumstances. But they did some sequencing of CBD and THC genes in several different varieties, and they showed that not only are there distinct versions of the gene, but that it looks like I hope you can read that the THCA synthase purple kush has one homolog up at the top, and then down lower, there's another homolog. So then the question becomes, not only is it not one, but it's two loci, but then do we have allelic variation in those genes, or do we have additional duplication events that have led to an even more complicated story? So we wanted to start getting at that, because if we want to develop genetic tools for identifying or predicting what plants are going to be producing, we really have to understand the genetic architecture a little bit. At least I feel like we do. So what did we do? We did a lot of sequencing. We have data for, hundreds of variants of these genes. But what we really couldn't tell by just the sequencing alone was whether, and several other researchers have tried this as well, is whether we're seeing allelic variation in the synthase genes or we're seeing multiple copies. And so we had this idea that we could use the flanking sequence on the 5' and 3' ends of the genes to try to determine if there are breakpoints that would indicate there have been gene duplications. And we thought, we can build this tool, this is going to be great, and this is a great idea. And then we find someone's already built the tool, which, in the end is really kind of cool because it works great. And I'm skipping ahead here. Let me just go back. So the idea here is that we can identify these breakpoints because even though you have a gene that's very similar, and it's so similar that you can't tell whether it's allelic variation or an actual duplicate, the breakpoints where a new duplicate would be inserted back into the genome would be non-contiguous, and you would be able to tell that it is a duplication. So there's a tool called GenSeed that actually has helped us to answer these questions. And it uses a hidden Markov model. It starts with a set of seed sequences. So we downloaded all of the available data from NCBI, and we used our own data to look at THC and CBDA synthase. And we actually worked with the author to develop the seed sequences for this model. And then what the tool does is it uses the seed sequence, so that synthase gene, and then it takes genomic data and tries to start assembling small regions that are flanking the seed sequence. And so the first set before the top blue bar would be these sequences that would align together and overlap the gene, and then you build a consensus sequence. And then you can build out a little further, and build a consensus sequence, and build out a little further. So we wanted to use this tool to understand cannabinoid synthesis variation in a worldwide population. And that's where we put into play our 96 samples of population genomic data. They're from, I think, at this point, over 90 countries. We have industrial hemp. We have land races that are really not cultivated varieties. They're from the point of origin. And then we have tons of drug cultivars from the US. And so the data that I'll show you today are from 8 different samples. We had deep sequencing on 3 different samples that are from 2 different subpopulations. Just mentioned that there are 6 subpopulations. And then we had lower coverage sequencing from an additional 5 populations. And so we pooled those data, assuming that they were similar enough to each other that they would have the same gene duplicates. And what we found is that there are 2 distinct versions, and these are, sorry, I should have mentioned this before. In the seed sequence that we use, these are only functional copies. So at this point, we have not assessed non-functional or pseudogene copies. So these are all functional copies that I'll talk about here. And so we found that there are 2 versions that have of CBDA synthase and then 2 versions of THCA synthase. In the data that we have, we've found that some of the samples, the one from subpopulation 1, sample 1A, has 2 copies of a CBDA synthase and no copy of a THCA synthase. And it's interesting because we know that this plant doesn't produce any THC. Now, this isn't definitive evidence. It doesn't have a copy of the gene because there could be some bias in the sequencing. So we see variation in the numbers of the synthase genes that we can identify. Some have 1, some have 2, some have none. And so when we look at the data that we have, we can identify a very clear breakpoint for these. This is just talking about the CBDA synthase genes. So the gray swath here is virtually identical sequence between the 2 different copies. And then once you break out of that identical sequence with, very minor genetic variation, you get into regions that are completely different, indicating that they are duplications. And then, let's see, yeah. We see the same thing for THCA synthase. We have several different samples that represent each of the 2 different copies. And once again, we see this very clear breakpoint. It's a little further out on the THCA synthase, but it's virtually identical within the gray bar and then completely different. I'm talking about the 5' end of this gene right now. I should have mentioned this as well. The 3' end of the gene, both of these synthases, is so similar that we weren't able to map outwards into that region with any fidelity to identify different copies. So, as have other researchers in the past few years, we support this 2-locus model for the synthase genes, but our data also suggests that this is not, that the variation that we're seeing within a variety is not allelic variation, but is due to additional duplications. And what we're doing right now is working with Jess's team to design primers so that we can validate that what we're seeing is not just some kind of computational artifact, but that we actually can design PCR primers and sequence them and provide further evidence that this is the case. We're also doing additional work to look at the timing of duplication events. And then the final thing that we're doing, if you'll remember, these were functional copies that we're looking at. And we know that others have identified pseudogenes of these synthases. And so I think we're going to add like another layer to this and build the seed sequence for GenSeed using the broken gene models, and then assess whether we're seeing even more copies that are non-functional. So I want to thank Dr. Gruber, who wrote GenSeed and has been really helpful in this project, and population geneticist colleague Matt Hahn, who was also a postdoc here, and then the science team at Phylos. This has been a project that that's been a whole team effort. And I think that's it.
Any questions? How big is the gray box and the duplication?
It's a few hundred base pairs on one of them and a little bit more on the other. I can get you the numbers, but
it's not hundreds of KB.
No. It's fairly short. Yep.
I was wondering, from your population study, if you have any data on the heritability of some of these chemical traits.
Right. We do not have that right now, but it's a great question, and we are working towards that. Yeah.
Can you talk a little bit more about the 6 populations and just what you're defining those as? It's kind of it's all based on.
Right. So we've, at this point, so we started, as Jess mentioned, with about 1,000 samples, and those spanned as much diversity as we could pack in 1,000 samples, land races from about 80 countries plus industrial hemp. And we looked at population subdivision. We did some pretty extensive testing looking at what is the right K, what is the right number of subpopulations, and this is using admixture. And so the K that performed best was 6, and it was consistent several years ago, as well as now that we have several thousand varieties and more countries represented. I just redid this analysis, and 6 populations is still supported. It's really interesting because we can see in the drug cultivars that there's a subpopulation that is a lot of the high CBD varieties, ACDC and Harley Sue, and some of those that are fairly well known, although I know names are difficult. And so we those come out together as a subpopulation, and we have some indication of phenotype being consistent. We also have a population of industrial hemp, the fiber type hemp, and that comes out as a solid subpopulation. And so there is some evidence that that these aren't just a computational kind of answer, that there are actual phenotypes that are assorting in these populations as well. The colors are.
Could you see an indica sativa population at all?
We don't. Sean Miles gave a really nice talk at a meeting I was at this summer, and he was talking about this and showed that there's a correlation between what people think of as indica and sativa and some genetic data and the phenotypes, the broad leaves versus narrow leaves. There's a bit of a correlation, but he really was warning against that as a useful means of identifying the effects that a plant would have. And in our work, we see the same thing. Yeah.
That's okay. So, have you found that the name that one person gave you doesn't align with somebody else gave you for the same genetics, or that the name is totally different genetics?
Exactly. So it's funny because Jess showed sour diesel. Yeah. And sour diesel, if you're in the cannabis industry, you've seen it, you've heard of it. It's fairly easy to fairly easy to identify because it's got a specific color, it's got a very fuely smell, and people are right about 50% of the time in identifying that plant, and that's an easy one.
Can I ask you a question that's related to that? I've been working on a different clonal population, a big collection for years and years, and it turns out a clone is not a clone, is not a clone. And you know, have you done any bisulfite sequencing or looked at the epigenetics that can be affecting some of that? And because these metabolic traits, so has that been done? Did you do the bisulfite sequence?
That is a really good question. We've been having conversations about what kind of experiments would really be beneficial for us to do, and we haven't done anything yet. But I will say that we have one cultivator that runs a nursery, and he has 3 varieties that he submitted to us, and they are genetically identical. They're all cuts of OG Kush. And we told him, and he's like, oh, that's great, that's cool, but they're all different. Like, I'm going to keep marketing these things as different plants because they look different, they smell different, they have slightly different potency. And so, you know, it's hard to know whether it's epigenetics or it's cultivation practices. You know, he already has a mindset that these plants are different. Does he treat them differently? Does he treat them the same? So, yeah, there's got to be some epigenetics at play.
A lot of times, though, like when people sequence and they have clones, they didn't realize they had clones. So our test in particular provides a lot of input to them to know you've been growing clones at the same time. Because usually they come back and they say, I knew it. I knew it. I just was too afraid to call them the same thing. And so, but
right.
There are the few who have seen indications of epigenetics.
Yeah. Primarily what we're seeing is what Jess is talking about, where they had an inkling, and we confirmed for them that they've got some redundancy in their catalog.
Quick question. So, it seems like it should lead up eventually to like GMO candidates, right? Is that what you're trying to do? Or
Okay, so one thing that was really exciting and maybe even surprising to us is that even though this plant has been in the dark for, how long has it been now, 60 years or some many decades, this plant has not really been grown publicly. We thought maybe that the drug cultivar set would have a really shallow gene pool, would be just not very diverse, and what we find is that there's huge diversity in cannabis. And what was your question again?
I just want to know, like, when are we going to get like clone material?
Yeah. So my point was that there's so much diversity in cannabis. There's been relatively very little breeding, mostly for potency, that there's so much that we can do without touching transgenics that it's just not really necessary. Yeah. Oh.
Go ahead. So I had a question. You had mentioned you have 1,000 samples. Is this purely just database, or do you have the actual germplasm that you're curating?
Yeah, so the initial samples were actually collected before I even got to Phylos, and those are usually little pieces of stalk. They're dead, dried up, so we don't have phenotype data. They're, just what we could either legally get in the United States, or we had DNA extracted in another facility, and then they sent us the DNA. So there's no collection really that's